So today I want to present I would like to present some of the studies that we have done over the years in the context of the effect of the environment and some viticultural strategies on the metabolism of the grapes, of the wine grapes. All right, so you see the pointer, perfect. So before I start, I'll try to keep the talk in half an hour. Before we start, I just want to give an introduction about where I come from and the viticultural region of British Columbia. So Miguel has introduced me by saying that, well, I'm Italian, I come from the northeast part of Italy, and it's where I graduated, University of Udine. But in 2013, I moved to University of British Columbia in Vancouver. And here is a map, a part of British Columbia. The Pacific Ocean is here, and Vancouver is about here at the border with Washington State in the United States. So we have 9 wine regions. Most of them are extremely small. I would say hundreds of hectares, but the total is 400. For some reason, you don't see the number, I think, on Zoom, on the slides, but there are 4,000 hectares in British Columbia. And this number, though, is increasing. The numbers of wineries has been growing exponentially. The industry started with what we call modern viticulture and knowledge in the 90s. And now we have about 320, 360 wineries. The 2 main producing areas, I will say, is the coastal area of Vancouver Island, but is actually of the 2 main, is a small one, is probably 20% of the production comes from here, but 85% of the production comes from this valley that is called the Okanagan Valley. And this valley is about a 5-hour drive from the coast, from Vancouver in the interior. And because there are mountains on both sides of the valley, it's a very dry region with 300 millimeters of rain per year. And it's quite warm. Actually, that's the Osoyoos here in the south is the warmest place in Canada. And in the summer, we hit 40, 42 degrees Celsius almost every year. We have warm summers, dry, but luckily we have these lakes that they run from north to south. And this is where the irrigation or the water for irrigation come from. In this valley that has, let me see, is warmer, I will say, than expected for Canadian standards. Is normally growing degree days is from 1300 to 1700, 1300 in the north part, 17 in the southern part. We grow grapes that are Pinot Gris, Chardonnay, Riesling, Pinot Noir, Merlot, Cabernet Sauvignon, and Syrah in the southern part of the valley. So we can really grow several varieties, several international varieties. In the island where it's more cool climate viticulture, we only have Pinot Gris, Gewürztraminer, Pinot Noir, and we still grow some hybrids. However, more than 95% of the grape production in British Columbia is Vitis vinifera. In Ontario, the main viticultural region of Canada is Ontario. They grow Vitis vinifera, but also several hybrids. We are more specialized on the vinifera. Long introduction, just a couple of pictures. This is Vancouver Island. Those are vineyards, as I said, cooler vineyards, close to the ocean. In this region, we do dry farming in most of the cases, or we can do dry farming. Most of the wineries are now installing irrigation because the summer gets quite dry. However, after having learned from Douro and other parts of Portugal, I'm not sure if I can say that it's very dry in the summer in Vancouver Island. But I can say that it's very dry in the Okanagan. This is a picture from the Inkamip Desert. Inkamip is the name that First Nation from the Osoyoos Band were calling that area. And it's a semi-arid shrubland. Basically, we cannot, again, I'm not sure if I can say that, but we have to irrigate our grapes, and we start irrigation sometimes blooming time if the spring is dry. Certainly without irrigation, we cannot reach the yields that the growers and the wineries are looking for. Today, in the final part of the talk, I'll present one of the projects that we did on irrigation in this area. But I would like to, go back and start my introduction now a bit about the effect of the environment on the grape metabolism. We know that the environment has a very strong impact on the quality of the grapes, obviously. And our laboratory have been working for several years particularly on management of water or water availability and the impact of water stress on the fruit metabolism. But we also work on the impact of temperatures. Those studies that I will present at the end are more related to greenhouse and growth chamber studies. And I will start by summarizing a bit what we know about the impact of water availability on grape or yield and grape quality. This is our review paper several colleagues and myself wrote in 2 years ago. And we pulled together many studies because we know that a lot of people have studied the impact of water deficit on yield and particularly on the phenolics of the grapes. So most of the studies focus on the phenolics. And you can see from this graph that, when we apply moderate or severe water deficit, we normally have a reduction of yield that obviously is larger when the stress is severe. We don't have large changes in sugars. We actually have some cases where sugar decreases and others where sugar increases under drought, depending on several factors. I think the genotype also play a role. Decreases in tetratable acidity, that is a problem for many areas, but it's quite consistent that people see increases in anthocyanins, and particularly in the concentration of anthocyanins, and some effects, but not as clear as in the case of the anthocyanins in the concentration of tannins. So overall, we kind of know that when we apply moderate to severe water stress, we have increases in the concentration of phenolics. Our laboratory, I'll present some of these studies, have worked in understanding if this was related to just concentration of these compounds that are produced in the skin of the berry, and or if there was actually a direct effect of the stress of this stressful situation for the plant on the biosynthesis of these compounds. And later on, I'll talk about the effect of water deficit on the production of other compounds like the terpenes or the aroma compounds that are more common to specific varieties. So I should acknowledge that several of the studies we have performed were in collaborations with different institutes, including the Edmund Mach Foundation in San Michele al Adige in Italy, Ben Gurion University in the desert of Negev, and the Institute of Applied Genomics in Udine. And the reason was we wanted to use techniques or platforms that allowed us to have large-scale and targeted analysis of metabolites and genes and the expression of the genes. And so the approach we had for these studies were what they are called, I guess, omics approaches. So we tried to do large profiling of metabolites and large profiling of the expression of the genes. Summarize and present only some of these results. But what we do, in the field is quite, is quite obvious. Also from this picture, we do not irrigate some plants like the one on your right, and we keep irrigating to optimal level vines. And so you see also the effect on the canopy. And then we normally collect samples for multiple times during the season and some cases intense, at several times during the season, from when the fruit is unripe to when the ripening process starts to harvest. And we profile metabolites and we profile transcripts to understand what is going on in a plant that is going under stress. I'm not going to present data, but normally when we do these studies, we also characterize the physiological response of the plant in terms of photosynthesis and stomatal conductance, etc. That I didn't include because we didn't, for a matter of time. So this is the result from one of our studies in Merlot. This study was performed by Stefania Savoy, who was a PhD student working with me at the University of Udine before I moved to British Columbia. But then she moved with me to British Columbia, and she helped me. We worked together in finalizing this study. So I'm not sure if I want to describe all this graph, but I will by saying that we profile in our studies primary and secondary metabolites from sugars, amino acids, and secondary metabolites like phenolics and volatile organic compounds in these grapes that were undergoing under stress. And we have all the data of which one they changed, which not. But what I wanted to summarize here with this principal component analysis, that both seasons where we applied the stress, 2011, 2012, we observed that by harvest time, that is this identified by these purple circles or squares, and when they are empty, it means that this is representing a stress sample. And when there is a full circle, is representing irrigated, or a sample coming from an irrigated vine. And what you can see is that regardless of the season, 2011, 2012, what we call metabolome of the grapes were similar amongst samples that have been stressed versus samples that have been irrigated. The main drivers of this difference were actually in these Merlot grapes were phenolics. We also observed some volatile compounds that they change in concentration, but main drivers were anthocyanins particularly, and also changes in stilbenoids like resveratrol, but those were actually decreases in our case. So I'm not gonna, these results have been published. I'm not gonna go into the details, but where we were interested was to understand if these changes we were seeing the metabolism were related actually to changes in the gene expression, so to changes in the pathways that are producing these metabolites. And we analyzed the transcriptome at multiple developmental stages. And here is the bars that you see in color means that identify the number of genes that were up regulated on this side, down regulated on this side, under water deficit condition at a certain fold change compared to the control. But take-home message is we found thousands of genes that were modulated. And interestingly, many of these genes were actually genes that belong to the pathways that were affected based on the metabolites by water stress. And this is an example of what we came up with. This was work of Stefania with Darren Wong, a bioinformatician who was very good in network analysis and is very good in network analysis. So we tried to put together to see if there was a relationship between these changes in gene expression and the changes in the concentration of the metabolite. And what you have here is phenolic acids. Those squares, diamonds actually, are metabolites that were modulated by water deficit. And if they are red, it means that water deficit was increasing the concentration. If they are blue, it means water deficit was decreasing the concentration. So we have phenolic acid, flavantriols, anthocyanins. So those are all metabolites that were upregulated by water deficit. And on this side, we had some stilbenoids like resveratrol or transresveratrol, PCH-ethanol. You cannot probably read the names, but those one were downregulated by water deficit in the same vines. And then all these dots are genes that belong to the pathway or to pathways that lead to the production of these compounds. And And the connection with red or blue light, it means that the expression of those genes were somehow related to the production of the metabolites we saw modulated. Basically, what we observe is that it was very consistent, the fact that genes were modulated and metabolites that are a result of those pathways were actually modulated accordingly. And this for us was interesting and important because often it is thought that water deficit do not affect the biology of the grape, but it's more like a concentration effect, as I mentioned before, what we see in the metabolites. So basically it's just a fact that the berry has less water and therefore the concentration of the compounds is higher. But with this study and other studies, we have shown that actually there is a direct effect on the metabolism. Another thing that is I think interesting to observe is that when we apply water deficit, we uncouple metabolites that are related to ripening. What it means is that, well, we know sugars and anthocyanins or aroma, they kind of increase, or some aroma, they increase during ripening. And here we plotted sugars versus anthocyanins to see what is the relationship under control conditions with irrigated plants. And here is under water deficit. And what we observe is that at the same, obviously, sugar levels, we have higher anthocyanin concentration. So it's not that by applying water deficit necessarily we achieve more sugar and therefore more anthocyanins, but at the same sugar levels, we have higher amounts of anthocyanins. I'll show later that for temperature, we have the opposite result. Probably already burned all my time, and then, yeah, no, okay. Yeah, but I have a long way to go. Okay, so, okay, I'll try to be faster here. This is actually a similar study. I want to present it. It's still Stefania's work, and because there is less work on the effect of lack of water deficit on white grape variety. And I think it's very interesting. I mean, I've been learning the past few days in Douro that, well, the white grapes are undergoing severe water deficit here in Portugal. I'm not sure if everywhere or in what region, but it's interesting because in many wine regions, people say you never want to stress a white grape because it's bad. So I don't know why it's bad, but they say that. And in our case, we wanted to study what's going on in a white grape that is going under water deficit. We did pretty much the same thing we have done for the Merlot study, have been presenting before. And here is a heat map of the secondary metabolites we analyzed. The way to read this heat map is, okay, here we have days after anthesis. Here we have the number for, from 27 to 93 days after anthesis was harvest. And each square, well, the core of the square identify if a metabolite was increasing or decreasing in concentration under water deficit. Red means increasing in concentration, blue decreasing concentration. Making the story short, we observed changes also in the white grapes, in the phenolics, but what we found interesting was that in these varieties, the name is Sauvignonas or Tokai Frulano, and several terpenes were increasing concentration at harvest under drought, or water deficit conditions. So we did sort of the same transcriptome analysis. I'm presenting here with a different graph, but when we mapped, basically we look at the genes that are controlling terpene production. We observed that actually they were upregulated. Several of them were upregulated by water deficit, consistently with what we observe for the metabolites. For instance, here is a graph of alpha terpineol, monoterpene, that normally in this variety is quite low in control conditions. But when the plant were severely stressed, these are very severe stress, it increased in concentration. And if you see the expression of some terpene synthesis, genes that control terpene production, well, it's also almost mimicking the same pattern. So we had a very interesting match between the expression of genes and accumulation of metabolites. And we have 3 terpenes, monoterpenes, that were increasing concentration. We thought this was interesting. However, I must say that this was a level of stress that our growers will not accept. It was a very severe stress. The canopy was, well, lost a lot of leaves. And again, I don't know, in different regions might have different opinions, but certainly our region will never accept this stress and this loss in yield. But another interesting thing is that Stefania and myself made the wines, and we found basically similar effects on the concentration of monoterpenes in the wines. And monoterpenes, for those of you that are not familiar with those compounds, are compounds, they have these floral or citrus-like aromas, and they are characteristic of, for instance, of Riesling, Gewürztraminer, Viognier, this type of, or Muscats. So those are important compounds for several varieties, not for all the varieties. But as I say, and when we had expert tasting the wine, they were observing differences in the aroma of the wine. So they were kind of matching what we observed by profiling the concentration of these terpenes. But as I said before, this was really a study that I'm not sure that if our growers, producers will endorse, because it was really severe. But when I moved to British Columbia, one of my first studies was to, together with Eugene Kovalenko, a master student, was to try to develop regulated depth irrigation strategies in vineyards to modulate terpenes. And we did this study in Gewürztraminer in the Okanagan Valley, as I said. Again, growing degree days 1600, so it cannot be defined as a cool climate region. It's actually a warm region. I think probably Bordeaux has around 1600, maybe now more, but definitely not cool climate. And it's an area that generally is dry, from May or June throughout the whole season. So we need to apply irrigation for that. The treatments we developed were treatments where we were irrigating according to the standards. We used leaf water potential to understand the plant water status and manage irrigation accordingly. Minus 0.8 is not, so these irrigation levels, I think they are like, they imply no stress, but at the same time, they are not very abundant. So it's in between what somebody will say mild stress or no stress. This is kind of the industry standard. But what we did was to apply regulated water deficit before veraison or after veraison or prolonged throughout the season. This results in different irrigation volumes. And if 100% was the control, with pre-veraison water deficit, we save 30% of water, late deficit, almost 40% of water, and with prolonged deficit, we save about 50% of water. This study was performed for 3 consecutive seasons, so we have some I would say long-term results. We profile several things from gas exchanges, sugars, and aromas. And I'm going with these results have been published a couple of 1 or 2 years ago. So if you are interested in any detail about the photosynthesis, etc., they are in the paper. But I'm just going to focus on a couple of data. The first thing is, depending when we apply the deficit, we might end up with different results in terms of loss of yield. Early deficit and prolonged deficit decrease yield significantly across the seasons. And so we have, actually, we have yields that I'm not really sure. Yields are, I was just curious after my tour in Dworo that yields are quite low compared to the ones we have here, but they told me that in other viticultural regions of Portugal, you have actually higher yields where you can manage irrigation. So in our case, the stress treatments reached 8-9 tons per hectare, and the irrigated one had 12.4, almost 12 also for late deficit. The take-home message here was that with late deficit, we didn't have a large decrease, actually a not significant decrease in yield. And this is, it makes sense because the physiology of the berry and the hydraulics of the berry changes from before and after veraison. And so when you apply stress before veraison, you end up affecting, and this is consistent with several studies, affecting the berry size much more than when you apply stress after veraison. Sugars, kind of the same story. Prolonged deficit was the one decreasing sugars. Again, this is bricks. Those are bricks, and this is the harvest of this winery. They want to harvest not too late because they care about acidity, so they harvest around 22 bricks. We had trials with other wineries where they go to 23, 24 according to their style. But since, well, there are decreases in titratable acidity, they don't want to wait too long. But you see the prolonged deficit decrease significantly the sugar level versus late deficit and early deficit did not. So this is the only figure that I present on terpenes, even though we have profile about 20 terpenes and at different stages. But this is the value of total terpenes, free terpenes at harvest. No significance, even though we have always the same trend across the 3 seasons for total terpenes, but significance for specific terpenes. We had 3 or 4 terpenes that were consistently increasing in late deficit, with late deficit. And among these 3 or 4, I mentioned 2 that are relatively important. They are quite important: geraniol and citronellol, because they affect the aroma of significantly affect the aroma of Gewürztraminer. So the point here is, again, free terpenes were modulated, and we saw that late deficit was the one that had a more consistent effect on terpenes. However, also the other treatments had tended to have higher level of terpenes, but never significantly, so we call it, no effect. Late deficit always had or had a consistent effect. In terms of bound terpenes, probably you're familiar with volatiles, and you know the several volatiles are attached to sugars. Those are not volatile compounds unless this sugar terpene bond is released through enzymatic activity or also some chemical reactions, and this could occur during fermentation and aging. But bound terpenes the trend was the same, but had no significant differences. So the difference we observed were on the free terpenes. Now, okay, I have 36 seconds for the 30 minutes, but you told me that I can do more than 30, right? Okay, I'll close my presentation with last 5-6 slides are about another program that is related, but kind of new in our lab. When we were doing these projects on secondary metabolites, phenolics and volatiles, we were observing changes in the metabolome. Those metabolites are very important for wine quality, so they are definitely a priority for our lab. But when we were analyzing this transcriptome from the Merlot study I published before, I did mention before, we observed that several of the genes that were modulated belong to the wax biosynthetic pathway. So the waxes are the wax that you find on top of the berry. And obviously the cuticular waxes are all on all the organs of the plant, leaf, berries, and they protect the organ from water loss, right? So they are very important component of the plant, obviously. Not sure how important for the quality of the wine. I don't think they are so important. That's probably why we don't study them so much. But we found very interesting that there was a modulation of this pathway, and we thought it was biologically interesting to follow up with that and to see if actually deficit was, or the plant was responding by producing more waxes to protect the berry from water loss under drying conditions. So this is a study that I present just one or two slides. It's a study that Nicholas Dimopoulos, a master student, did in my lab. And he performed this greenhouse study where he applied severe water deficit to Merlot grapes, collected berries, and analyzed berry for the wax content, wax profile, and also he did electron microscopy to see the structure of the waxes on top of the berry. And I'm presenting just 2-3 graphs, the total aliphatic waxes and triterpenoids, 2 components of the waxes of the grapes. We observed an increase in the production of waxes under water deficit that was consistent with the gene expression data we had from before. And interesting enough, also the structure of these waxes was changing. Here is a control. I'm not sure how well you can see from there, but the control have these, I don't know, leafy style waxes. But when we were applying deficit, we observed with obviously repetitions of the analysis, replicates, that the waxes become more like ribbon, I don't know, like this thin, leafy or algae style of waxes. So we observed changes in the structure. So we thought, okay, something is happening. And we performed the analysis of water loss from the berry. Like we analyzed the water loss from the berry during under water deficit from early stages of development to harvest. Here I'm presenting only harvest. And we did observe less water loss from water deficit. So a trend toward less water loss from deficit berries. However, well, first of all, it's almost significant, but it's not. Well, then it depends what are statistics for you. However, even when we calculated the kind of the amount of water loss, it was kind of marginal. So if there is a change, the change is marginal. And our conclusion was, yes, we do see this response of waxes, but this response of waxes is not really, we don't think that is affecting the water loss from the grapes. Waxes also respond, well, are important for other things, including defense from pathogens. And interesting enough, we do see less under deficit. We did observe less pressure from some pathogens like powdery mildew. But I will not. There are many reasons why this could happen, so I don't want to go there. But the story about the waxes and water deficit is we definitely see a change in the waxes. But our theory was that it was helping preserving the water in the berry, but we don't see major differences. But and now I'll really rush to the end, is the waxes come back also in other studies we are doing now about temperature. As I said before, we work on temperature and we try to assess the effect of temperature on metabolism. This is just a review from literature that I will actually skip. There are many papers talk about how temperature or a very high temperature might affect the berry. There is a general, well, we know that with higher temperature, we have more sugars, but we also know that very high temperature, like extreme temperature, like 40 degrees, might block the sugar accumulation, might block the berry growth. And we did some work on the anthocyanin pathway and to assess what were the optimal temperatures in Merlot grapes using growth chambers, but also to assess if it was the day or night temperature or difference between day and night temperature, because it's a common belief, or maybe it's right, I don't know, we always say is the delta temperature between day and night that affects the anthocyanin production. And this is, kind of, I will say, well-known viticulture enology, but not really supported by our data. The thing is, at least in our experiment, we put plants in the growth chambers. We studied 35, 25, and 20. 1 year, another year was 30, 25, and 20. And we tried to play with this night temperature and day to have gaps of 10 or 15 degrees Celsius, and to see if this between day and night, and to see if this was affecting that. Probably these gaps are not as big as others that we observe in vineyards. So we can, our data are, well, probably limited compared to all the possibilities you can have in vineyards. But in our study, we observed this thing that temperature was definitely negatively affecting anthocyanins. So the highest values were with plants at 20 degrees Celsius and the lowest at 35 degrees Celsius, and 25 was really intermediate here. But when we were playing with day and night, we didn't see a big difference between, what we observed was that it was the daily or the average, the daily temperature was ruling. And we had very similar results between, for instance, plants that were at 20 degrees and 10 in the night, 20 in the day and 15 in the night. And the same also with the 35 ones. So, and the next year, the same thing. So our observation was that the delta temperature between day and night did not have such a strong effect like we were hypothesizing, actually. And it was more the daily temperature were affected the level. But again, as I showed before, an uncoupling effect due to a temperature that is opposite than water deficit. So at 20 Brix, you have low anthocyanins with a high temperature and high anthocyanins with low temperature. Now, I just rushed to the end. I think this is an interesting point of discussion after I also saw I think particularly in Portugal, because I don't know much about Portuguese wines. However, I drink them and I find they always have a nice color. And I find, very dark, intense color. I know you use also some varieties that help with that. And some varieties are rich in color. And I know you have very, how to say, small berries and very concentrated. So that's part of it. But definitely high temperature are not helping with the production of anthocyanin. I will really close here with 2 slides. I'm sorry if I'm taking too long. We started working on heat waves or effects of extreme temperature for a few days on the physiology of the grapes. And we just published this paper where we applied in growth chamber single and double heat waves that we are experiencing in the vineyards. So those numbers, 40 degrees, are happening in our vineyards in British Columbia. And those 30, that is the control, is sort of where they normally grow the plants during the summer. But when we have this heat wave, we have a few days around 40. So we applied these cycles of 4 or 5 days in the growth chambers where we were exposing plants to a heat wave, simulating a Pacific Coast heat wave. And our focus, the study was on the physiology, actually, photosynthesis, etc. But Josh Vander Weide, who is now professor of Michigan State, wanted to see the effects on the waxes. So I'm connecting again with the waxes, and he observed that actually double heat waves were strongly affecting the waxes, increasing the production of several waxes, from the total waxes to the triterpenoids, and decreasing wax esters. So waxes have different categories based on their chemical structure. And we found that was interesting. So he performed this experiment similar to what we did with the water deficit one, to see how much water we're losing. And we actually observed this time that when there was a strong effect on the waxes due to double heat waves, the berries were losing less water. And that was also interesting because we observed that exposing plants to a single heat wave will be decreasing the berry weight. But when we expose grapes to double heat waves and we have more wax production, we had no changes in the size of the berry. So we think that unlike what we observe water deficit, this time we see an effect probably on the water loss of the berry that might mitigate a severe stress. This wax thing, I don't know how interesting it is for people, but I work on also on blueberry. Blueberry is a major crop in British Columbia and Washington State and Oregon. And we had heat waves. I'm not sure if you heard in the news. Well, we have heat waves everywhere, so it's not from the news anymore. But 2021, we had big heat waves in the Pacific coast. And I was attending a talk, and this person talking about blueberries was saying that they observe a much different bloom or wax on the top of the blueberries under those conditions. So I think that this is a topic that is of interest of our lab, and in the future we will work more on this. That's why I wanted to present a bit about that. Well, I'll go close to the conclusion. My conclusion of this different study is that water deficit is a direct impact on the metabolism of the berry. It depends when we apply water deficit, we can have different results. And the temperature sure has an effect as well. In our study do not indicate that the delta is such a critical effect, delta day-night temperature. And then the waxes of the berries are responding to environmental stresses. How important for the final quality, not sure, but probably it's important for the berry response in terms of water loss or adaptation to the stresses. I would like to thank many students and collaborators that participate to this work, as well as funding agencies. I've been presenting studies from from 2015 to now, so there are many people I have to thank, but particularly I would like to thank you for your attention. I'm sorry if I probably went too long. Thank you.
So I will leave now this part to some questions from the audience or via the people that are on the other side, of course. So maybe here, is there any question?
No.
Okay. I had a question. Actually, I have two questions. The first one, and they are both about the waxes. The first one is, you were saying about the double heat wave. And what was the difference in the timings of the heat wave? When was the single one, when was it, and the double?
Yeah, so the single one corresponds to the second heat wave of the double that is during ripening. So after veraison, just after veraison. The double treatment had a heat wave before and after veraison.
Okay.
Before, so one is, there is about a difference of, I think, 15 days, 20 days between the two heat waves, but one was applied before the beginning of ripening and the other after the beginning. Sorry, yeah, after veraison, yeah.
Okay, thanks. And the other question was, it's related to what we were talking before. So I did my PhD on the image analysis for yield estimation, and actually there are some references where they collect pictures of the grapes at a certain stage where there's no wax yet.
Yeah.
And they use artificial lighting, so that because there's no wax, with the lighting they can detect the berries better. So I was just wondering if this effect of the water stress also affected somehow the timing, or was it of the waxes, or maybe it could impact this methodology?
It's a good question. I will disagree that there are no waxes before. I think there is so I presented, I mean, I'm just saying, because I agree that there is a change in the wax amount and profile. So there are different compounds. So we are, making the story short, in this paper, we analyze waxes from early stages of development, like 30 days after anthesis, and then, you know, until harvest, right? And so you see a change in the amount of waxes, but also a change in the type of, in the profile of the waxes, so the relative abundance of the different types. And if you actually analyze also the water loss, there is a change in water loss from high water loss earlier on to lower water loss later on. So the waxes are always there. They change in amount and composition. Yes, the impact on the phenology, right? Is water deficit just accelerating some process of ripening? It could be that is somehow an acceleration going toward a certain stage of that you have later during the season. But if you see, for instance, other parameters of ripening, they remain consistent. So it's always difficult, and I don't want to take too much time to figure this out, if it's an acceleration of a process or if it's a direct effect. But the fact that we see changes in gene expression, the fact that we see no changes in sugar, but then changes in waxes, you know, let us thinking that it's a direct effect on the wax.
Thanks.
Yes. Oh,
A professora Elizabeth tinha pedido, eu posso já.
I would like to thank you for your presentation, because I'm not at all expertise, is not my research area. Mine is environment engineer, but you are so clear that it's very important to the message, and I'd like to congratulate for that. Even I'm not research in this area, but because I have worked with Miguel Costa, I would like to have two questions. The first one, because you talk about availability of water, yes? And in my opinion, the first question is, you mentioned the stress and the things, but I'd like to put one question. In 1 year, rains quite a lot, can happen. And the other, like happened in Portugal, it's very dry. Can you model it at least? It's not possible, you know, because you need to anticipate what are you going to do. I just stay or I do to irrigate. What is the compromise between these two things? Availability of water and yield. This for me, because I'm looking about what to do with the water, because I'm more concentrated in wineries and the wastewater treatment, recycling this water, all this compromise. What is the decision?
Well, so, okay.
Thank you for the clarity of the thing,
Oh, thank you.
and thing.
Thank you.
Thank you very much.
Okay.
I learned quite a lot, even though I'm not in the area. Okay.
Good, good. Thank you very much. So to answer, let's see, specifically, I will say that, yes, it depends on the wine region, right? There are wine regions where droughts are quite consistent. So at the end of the day, and you have water available. So at the end of the day, it's just a decision of the grower if to apply this water or not. And so you can take these studies in a way that is, okay, let's look what is the best thing for the quality of the grape. Okay, so that could be one goal of the studies. And this is under the assumption that you basically can control always, because like in Okanagan, they have to irrigate every summer and you have water available. That is a concern also for the Okanagan Valley, because the Okanagan Basin that is reaching water is forecasted to be in deficit of water because the urban areas are growing. And so this all agricultural land that uses a lot of water will have to compete for water because they have the same sources. So it's a very important topic. But this is where I think that that's why also we study these things, to understand how much we can push a grape, until we see changes in yield or changes in quality that might compromise your or affect your income. So I'm saying that I agree with you that there are different scenarios or different things we have to take into consideration. But regardless, I think it's important to study this so we can somehow we have more information to respond. Yeah. In the past, when and I'll conclude, probably many years ago in many wine regions where there was water, they they were focusing on yield, so water was very important, and we were over irrigating. Now that we know that yield quality could be penalized, now there is more attention. I'll also say, managing carefully in the water, that is also good for the environment and the water and the preservation of water resources. Yeah.
Okay, thank you. I'm very happy to have you in person since the time I read you. And the question is about something that you didn't mention. I'd like to ask you if you look to the barrier, cell wall.
Oh, the cell wall.
First. Second question. Are you so confident about greenhouse studies compared to the field? Because the root system is always something that we cannot compare. Thank you.
Okay, good. So 2 difficult questions. Let's see how the first one. The first one is no, we did not work on the cell wall specifically. We did observe in the transcriptome studies that underwater depth we saw modulation of some cell wall genes. I think there were pectin methyl esterases and expanses, but this is stuff that I go by memory, because in another study we see a lot of those modulation, but it's because it was focused on ripening. But I think, if I'm not mistaken, ABA, that is a hormone that responds to water depth, is also a modulator of cell walls genes. So there might be an effect of this, but we didn't focus on our analysis on those genes, so I cannot answer with clearly on that. The second question, is sort of like there is no doubt that we have to be careful in what we do when we start. Well, when we use a control environment for our studies, right? Because the plant is different. There is no root. The root system is confined in 10-liter pot. However, the field studies present other challenges. For instance, in the temperature situation, we do the control environment for temperature. The concern in the field is that it's almost impossible to uncouple temperature with light, and we know both factors are very strong. And so, the answer is yes, probably it's not the best case scenario, but is a scenario where sometimes is the possible one. I will just conclude by saying that no, everything is for sure related, right? Roots and in particular, how we can buffer some environmental condition, the plant, not we. But not bad, but maybe I should stop here. What I wanted to say is that we believe that a lot of things happen in the berry. The metabolism in the berry is very sensitive to the environment itself. Definitely is connected with the water arriving from the root and the sugars arriving from the canopy. So everything is connected. But my impression is that some metabolic patterns are very sensitive to the condition of the organ itself. Probably this is where these studies work well, but when we put in the context of the whole physiology of the plant, yes, it's a very different condition.
Thank you also for a nice talk. Simon, I was wondering, this increase in the free terpenes that you noticed, did you also see in the transcriptomics, like the downregulations of genes coding for enzymes involved in the glycosylation of these volatiles, something like that?
Yeah, good question. What we observed was the glycosyl transferases that they glycosylate them. Mm-hmm. We had a couple of them were actually upregulated as well. So the ones that they promote attaching the sugars were either not modulated or positively modulated, depending on the experiment we have run and the stage. And I think in the paper we are presenting an expression level of one of these glycosylases. Then these theoretically attach the sugar, right? So if there are other enzymes that contribute to in the berry in these in the function of breaking the link, we don't know. Yeah, we were not aware of which enzymes to look for, and we haven't observed that. But it was something that we thought about. What happens to this balance between free and bound? I must say that we observed kind of both of them increasing. So they, as I said, one was not significant, but the trend was similar. Like the graphs, they look quite similar. So probably it's not significant because we have a lot of noise and variability, and we need more replication. But yeah, no, we don't have that those data on enzymes that will reverse the link.
And just also curiosity, because of some data that we also have, did you also notice increase in ethanol in these samples? Ethanol.
I think, okay, oh my. This brings, in the samples in the berry.
Yeah, well, the ones submitted to the the ones submitted to the water stress.
I think, so the reason I'm responding like that is because I cannot say anything, because now I don't think it relates to these ones I showed, but during my PhD I did similar experiments. And I think in one analysis I did observe an increase in ethanol. But this is, and we never paid too much attention to that, honestly, but yeah, was from my studies in 2007. So now I cannot, I'll have to go back and see. But when you mentioned, I kind of remember that there was something like related to ethanol, and I think it was a slight increase, but I cannot, we never follow on that. Never analyze more in depth.
Mais alguma pergunta? Any other question? Well, I will ask one, mostly related with the boxes, because Gonçalo, we mentioned a part of light. Maybe, Simone, the optical part of the berry in relation to light can be interesting, because the reflection. So maybe boxes can also have that influence and eventually affect the berry temperature. That is a major issue. So maybe that part can be interesting. I want. Other thing that we were also, and here in ISO, we also interested is the impact of soil temperature on berry temperature, and the experiments done in lab. Maybe they can separate that two issues. And the other, finally, one, just to finish, right, is shall we proceed with experiments in more extreme conditions rather than softly extreme? So I mean, no water, higher temperatures. What is your expectations? What should, because regarding what we are experiencing here in our country and other Mediterranean countries, we are talking about extreme, and more extreme conditions. So.
Yeah, this is a very interesting question, because okay, my experience was always depends on the study we run. Sometimes we want extreme stresses to see an effect, a large effect, and try to figure out what's going on. In other studies that normally are more applied, we try to simulate what growers tell us that is the issue or could be the issue. Interesting enough, the heat wave experiment I showed at the end, that is one of our last experiments. I told my postdoc, "You are crazy. In Apply 42, they will tell us that in British Columbia we cannot have this." They will criticize why. Right? And actually, one reviewer say, "Why you choose that?" Luckily, or actually not luckily, unfortunately, the heat wave that we have in British Columbia now are 42 degrees. I'm just saying, to your point, what in the past I would have thought is an extreme condition that will never happen in a real case scenario in our viticultural region was actually the real case scenario, right? So I think, well, I don't know if I have an answer, but I think, yes, we have to keep experiencing these things. And yeah, we have in general two approaches. Sometimes we're very extreme, but as I say, now what I consider very extreme is a real case scenario. And sometimes we really try to focus on what the industry tell us that is their scenario and simulate or try to analyze that type of scenario. Yeah.
So thank you. Don't know if there is any other question. So I think we must finish now, and thank again Simone to be here with us in Lisbon at Instituto Superior de Agronomia.
Yeah.
And I must thank really the opportunity that you have gave to come to us. Okay.
Thank you very much.
Thank you.